I'd like to welcome An Hendrix. She's an assistant professor at the Laboratory of Experimental Cancer Research, Ghent University, and at the Cancer Research Institute Ghent, Belgium. So Anne is well known for multiple high-impact papers that dissect methodologies for EV separation. She has also developed the EV track knowledge base, which are very well recommended, and also protocols and reference materials to improve transparency in EV research. So Anne is also known as the organizer of EMBO hands-on courses. Today, she's going to give a presentation on the biomolecular landscape and dynamics of EV in blood plasma. So welcome, Anne.
So first of all, Carolina, Jan, and Dolores, I want to congratulate you with this initiative because I think it's a very valuable source for the EV research field. And I hope that with this presentation, I can add another valuable talk to this series. So the aim of my presentation today will be twofold. So first of all, I want to highlight some of the pitfalls and challenges that occur when you're studying extracellular vesicles in complex body fluids or biofluids such as blood. And after the methodological challenges, I will further move in towards the biology. What can we learn from extracellular vesicles in blood samples from patients with different types of diseases? Okay. So first of all, if we study extracellular vesicles in blood, we have to take into account the complexity of blood. So blood contains blood cells, platelets, abundant proteins and aggregates, cell-free nucleic acids, but also lipid carrying particles, including extracellular vesicles and lipoprotein particles. So if you want to study the biology, function, composition of extracellular vesicles in blood, we need to be able to capture them out of this complex mixture. And this is mostly hampered by proteins and lipoprotein particles that share biophysical characteristics with extracellular vesicles, such as size and density. Now, there are multiple methods available to separate extracellular vesicles from biofluids. Some of those methods, including differential centrifugation, size exclusion chromatography, and ultrafiltration, separate extracellular vesicles based upon their size. Density gradient centrifugation exploits the density of particles to separate extracellular vesicles. Immune capture makes use of antibodies to target surface antigens that are available on certain extracellular vesicle subtypes. And then there are also some precipitation-based methods that basically extract biological material from a sample that you are studying. Now, of course, one of the first key questions is whether it matters which method you implement to separate extracellular vesicles from your biofluid of interest. And given the majority of methods and protocols that is available, this is probably one of the first questions that many researchers that enter the field encounter. And this was also the case for my research group. So before we embarked into studying extracellular vesicles in complex body fluids like blood, we decided to set up a standard experiment or a proof of concept experiment to test whether the choice of separation method influences the downstream analysis or identification of the content of extracellular vesicles. So we started off from the most simple biofluid, cell culture supernatant. And we prepared cell culture supernatant from MCF7 breast cancer cells. So there are a few pre-analytical steps that we implement, like low centrifugation speeds and filtration to separate cell debris and also some free-floating cells. And then the concentrated cell culture supernatant was divided over 3 commonly implemented methods. The first method was differential ultracentrifugation, where we pelleted the extracellular vesicles at 100,000 G for 3 hours. A second method included the OptiPrep density gradient centrifugation, where we load decreasing concentrations of iodecanol on top of each other. And on top of this gradient, we load the cell culture supernatant. This gradient is then centrifuged for 18 hours at 100,000 G, and then we collect the different density fractions from the top to the bottom. The third method that we included in this comparison is the commercially available kit ExoQuick. Now, ExoQuick exploits polyethylene glycol to precipitate biological material from a sample. The advantage is by using PEG that we only need low-speed centrifugation steps to pellet down this material. So we prepared extracellular vesicles from the cell culture supernatant using 3 methods, and we implemented complementary characterization methods to characterize those preparations in depth. I will not go through all of them, but focus on the RNA analysis that we performed. And what we could actually observe is that the RNA signature that we identified in the preparations that we obtained by the density gradient differs from the RNA composition that we identified in the preparations that were prepared using ExoQuick or differential ultracentrifugation. So this 6 years ago was a striking observation because we did not really expect that the choice of method would influence the downstream analysis such significantly. So then, of course, the question is, how is it possible? And of course, it's very likely that each of those methods separates extracellular vesicles with different efficiency, with different specificity. What do we mean with different specificity? Not each method will only separate or enrich for extracellular vesicles, but can also co-separate or co-enrich other extracellular particles. For example, ribonucleoprotein complexes. These complexes are known to circulate microRNAs independently from extracellular vesicles in blood. One of the core proteins of ribonucleoprotein complexes is the Argonaute-2 protein. So we evaluated the EV preparations that we obtained for the co-separation of the extracellular vesicle-associated protein TSG101 and the Argonaute-2 ribonucleoprotein complex protein. What we observed is that with density gradient centrifugation, we have a clear enrichment in the extracellular vesicle-associated marker, while with differential ultracentrifugation and the ExoQuick method, we had a clear co-separation of both the ribonucleoprotein complexes together with extracellular vesicles. One of the nice things about the density gradient, and I will come back to this later on, is that you can actually study the different fractions of the gradient. And this is what we did here. So we collected the different fractions from the top to the bottom. We refer to these fractions from 1 to 14. What we observe is that extracellular vesicles float at their typical density of around 1.1 gram per ml, whereas the Argonaute-2 ribonucleoprotein complexes were retained in the top fractions of the density gradient. So this indicates that by using density gradient centrifugation, we were more able to separate different types of extracellular particles, increasing the likelihood or that we were identifying extracellular vesicle-associated RNA content. So this is, of course, a very important consideration. Depending on the method that you will use, we will enrich extracellular vesicles with different levels of specificity, and this will, of course, induce the identification of method-dependent extracellular vesicle composition, but also extracellular vesicle function. Now, given the importance of the methodology, we were next wondering what the heterogeneity is in methodology that is used in extracellular vesicle research. So we decided to do a literature study of publications that reported on extracellular vesicles between 2010 and 2015. We selected 1,200 publications that were analyzed for 115 experimental parameters. These experimental parameters were related to the separation of extracellular vesicles, also their characterization at the protein level and at the particle level. Now, there are two important conclusions that I would like to share here. So first of all, for the 1,200 publications that we investigated, more than 1,000 unique separation protocols were reported, meaning that there is indeed a big heterogeneity at the methodological aspect. Furthermore, almost 20% of those studies did not characterize the extracellular vesicle preparations that they were studying. Given this, it's important that we have to consider that there is methodological heterogeneity, but we also have to consider that this methodological heterogeneity will likely not be solved over time, because depending on the biofluid that you are studying, depending on the aim of your study, we would need different methods that could be combined in one or another way, but we need a diversity of methodology to fully explore the potential of extracellular vesicles. Nevertheless, if we use different types of methods and protocols, it's very important that we transparently report on these protocols so that compositions, biomarkers, functions that we identify at least can be reproduced by other researchers in the field. So based upon these pioneer studies, we decided to focus and put some effort in developing a supporting ecosystem on how we can increase reproducibility in the field, because this will be key to translate our know-how of extracellular vesicles into clinical applications. So a first effort that we made was to develop the EVTrack knowledge base, which is an online toolset for the transparent reporting and also centralizing knowledge on extracellular vesicles. Now, what is the key aspect of this knowledge base? Actually, it's twofold. On the one hand, it's aimed as a coaching tool that can stimulate or help researchers in transparent reporting. But on the other side, it's also a knowledge base, capturing information that is not searchable to other web engines like Google or Web of Science or PubMed, for example. Now, how should the EVTrack knowledge base be used in case of increasing transparency? What we aim for is that once you're at the stage of preparing your manuscript for submission to a journal, that you upload the experimental parameters of your material and method section to the EVTrack knowledge base. In return, you will receive the EV metric, which is a measure for the level of transparency in the material and method section. Based upon this EV metric, you can increase the transparency if required. So it's a sort of coaching tool that helps you to assess which are really the important experimental parameters that can influence the sample that you are preparing, and thus also the identification of functions and biomarkers. By doing this, we of course, as a knowledge base, are able to capture how the field is evolving in terms of methodological aspects or new characterization methods popping up, isolation methods. And this is important to also keep track on the evolution of the extracellular vesicle field so that we can maintain minimal information guidelines up to date. Once the publications or the manuscripts have been accepted for publication, the information that was uploaded to EVTrack will be publicly available on the EVTrack knowledge base and can be searched by users for relevant information. It's also possible to, if you were not aware about EVTrack or you forgot in the submission and exception process of the publication, that you add additional information post-publication. So you can still submit your experimental parameters post-publication. And if there are some parameters that you are now aware of to be important, but that you did not report in your publication, you are able to add this information to the EVTrack knowledge base. Now, how does the EV metric look like? It consists of 9 experimental parameters that we, with the consortium, concluded to be important to have an adequate interpretation of the EV preparations that have been studied in a publication. These correspond to the MySEF guidelines from 2014 and will soon be updated with to the new MySEF 2018 guidelines. But basically, if parameters have been adequately reported in the publication, they appear in green. If they have not been reported, they are indicated in red. So it very straightforward informs you on which parameters should also be reported in your material and method section. And actually, in 81% of the experiments, it's so that an increase in transparency is achieved by increasing reporting rather than performing additional experiments. Now, the EVTrack knowledge base has been endorsed by the International Society for Extracellular Vesicles, and it's now also one of the recommendations of the MySEF 2018 guidelines. So transparency is, of course, one important aspect, but it would still not solve all reproducibility. It's the first step forward if we are able to reproduce each other's experiments, but then we can still have inter-user variability, variability in between labs, between different patients, and within a lab, even between different time points. So to solve and to further increase this reproducibility issue, reference materials will be key. A reference material can have multiple purposes. They can assist method optimization, also method optimization for extracellular vesicles. They can help to identify which pre-analytical variables are influencing the identification and characterization of extracellular vesicles, such as choice of blood collection tubes, storage temperature, transport, and so on. It can also help in calibrating the measurement methods that we use to quantify the number of extracellular vesicles, and it can increase the reproducibility of the results. And of course, it can assist in data normalization so that we are able to mitigate for technical variability that is measured between different samples to reveal the true biological heterogeneity. So to address this need, we decided to develop recombinant extracellular vesicles as a biological reference material. How did we develop those recombinant extracellular vesicles? So we basically overexpress the HIV-1 GAG polyprotein in HEC293 T cells. This results in the production of extracellular vesicles that have that really resemble endogenous extracellular vesicles that we encounter in different biofluids. So we have demonstrated that those recombinant extracellular vesicles, first of all, highly express the GAG protein that is fused to the EGFP fluorescent protein, which results in highly fluorescent extracellular vesicles. Furthermore, we demonstrate that they resemble endogenous extracellular vesicles at the lipid level, but also at the protein level. So we identify the common surface proteins like different tetraspanins, but also some luminal EV-associated proteins like LX, DSG101, and syntenin-1. Furthermore, these recombinant extracellular vesicles share both their size and density with endogenous extracellular vesicles. Now, important for the broad application of a reference material is that, of course, it has multiple readout options that are available in routine laboratories. We demonstrated that based upon fluorescent measurements of the GAG-EGFP protein, that recombinant extracellular vesicles can be quantified by making use of a fluorescent parade reader, by fluorescent nanoparticle tracking analysis, and also by high-resolution flow cytometry. Furthermore, recombinant extracellular vesicles can be quantified for their EGFP messenger RNA by qPCR analysis and also by ELISA based upon the GAG polyprotein using the P24 ELISA assay. So what is then the applicability of those recombinant extracellular vesicles in the EV research field? Of course, it's multiple-fold, but here I focus on two examples. So first of all, if you spike those recombinant extracellular vesicles in your biofluid before you do any preparation step, you are able to mitigate for inter-user variability. So here we have an example where we have no, in the absolute condition, where we did no spike of the recombinant extracellular vesicle and no normalization of the data. However, if we do it, the same experiment with multiple users that are implementing a separation method to obtain extracellular vesicles from a biofluid, and we can take into account the recovery efficiency of the recombinant extracellular vesicles that we spiked into this biofluid, we observe that we can reduce the variation with 66%. Furthermore, for example, if we separate extracellular vesicles from the blood of healthy donors versus cancer patients, and we do not mitigate for technical variability, we did not observe a significant difference in the number of extracellular vesicles in the blood sample. However, if we spike the recombinant extracellular vesicles before we separate them and then quantify the recovery efficiency of the recombinant extracellular vesicle and normalize the data accordingly, we observe that we do identify a significant difference. So these are two applications of the many that are available to apply extracellular vesicles to mitigate for technical variability and normalize data. Okay, but how do we then actually study extracellular vesicles in blood plasma? Of course, we also need a standardized operating protocol procedure to do this. And then again, we have to take into account the complexity of the blood. This is a very nice schedule that was published by Simonson in 2017 that really maps how extracellular vesicles overlap partially in size and density with different other types of extracellular particles. For example, extracellular vesicles overlap in their density with high-density lipoproteins, and they overlap in size with protein aggregates, low-density lipoproteins, very low-density lipoproteins, and kilomicrons. So from this schedule, it is clear that if you would combine a size-based method with a density-based method, that we might be able to obtain a highly efficient, specific separation procedure for extracellular vesicles. So what we implement is that we start from 2 ml of platelet-depleted citrate plasma. We load this plasma on a size exclusion chromatography column, and we collect the fractions that contain the extracellular vesicles. We refer to this fraction as the crude extract. The crude extract is in a second phase loaded on top of an OptiPrep density gradient. This density gradient is centrifuged 18 hours at 100,000 G. And what will happen is that the different extracellular vesicle particles will flow to their respective buoyant density. So theoretically, from this schematic, we would conclude that most of the HDL particles would be removed by size exclusion chromatography, and that by making use of a density gradient, we will remove the remainder of the low-density lipoprotein particles. Now, there are two important aspects to this fractionation schedule. First of all, size exclusion chromatography is commonly implemented as a standalone method to separate extracellular vesicles from blood. So what we decided to do is to perform an in-depth analysis of the protein composition of the crude extract compared to the extracts that we obtained from the density gradient, namely the EV extract and the lipoprotein particle extract, or the LPP extract. Secondly, since we do this fractionation, we can broaden our scope and not only focus on the biomarker potential of extracellular vesicles, but also study the biomarker potential of LPP lipoprotein particles and see whether there is an overlapping capacity in biomarker potential or if they have an added value on top of each other. So we optimized this protocol we collected the crude extract, the LPP extract, and the EV extract, and we performed a mass spectrometry-based proteomic analysis. From this hierarchical clustering, we could conclude that the protein composition of the extracellular vesicle extract significantly differed from the protein composition of the crude extract, which is the extracellular vesicle preparation obtained by the size exclusion chromatography only. More strikingly, actually, is that the crude extract corresponds more to the LPP extract than to the EV extract, which already is a first indication that we can obtain more information by getting a more straightforward and more specific separation between extracellular vesicles and lipoprotein particles. Furthermore, this principal component analysis demonstrates that actually the methodology, although it's quite complex to combine a size exclusion with a density gradient, is highly reproducible and is in a similar reproducibility range or repeatability range as size exclusion chromatography. Furthermore, the principal component again demonstrates that the extracellular vesicle extract significantly differs in its protein composition compared to the crude extract and the LPP extract that correspond to one another in the first component. If we take a further in-depth look into the proteomics data that we obtained, we can observe by searching the reported HDL and LDL proteome that has been established by the Cha and Davidson labs, that in the LPP extracts, we see 55% of the proteins are actually HDL associated proteins and 40% of those proteins are LDL proteins. So in the LPP extract, we see an enrichment for those lipoprotein particles compared to the crude extract. And furthermore, we observe that these proteins are significantly depleted in the extracellular vesicle extracts. This is further confirmed by the electron microscopy, where we observe in the crude extract a mixture between some extracellular vesicles and different types of lipoprotein particles. After the density-based separation, if we take a look to the LPP extract, we don't identify extracellular vesicles, but we do observe kilomicrons and different types of lower density lipoprotein particles. And in the EV extract, we see a clear specificity towards those vesicles that are depleted of lipoprotein particles and protein aggregates. So setting up this strategy, demonstrating the repeatability and showing a significant difference in the protein composition between LPP and EV, we further decided to study the biomolecular landscape and the dynamics of extracellular vesicle in blood plasma from patients that have been diagnosed with different disease. And we did not only focus the study on extracellular vesicles, but decided to compare the performance of extracellular vesicle extracts to lipoprotein particle extracts. So the patients that we included in this study were a total of 38 samples, and those patients were diagnosed with breast cancer, ovarian cancer, or HIV. We prepared the LPP extracts and the EV extracts by implementation of the orthogonal implementation of size and density-based separation. And we performed a mass spectrometry-based proteomic analysis of those preparations. First of all, for the extracellular vesicle identification, we observed that a variety of extracellular vesicle-associated proteins were identified across the different disease conditions, and that we have a significant number of extracellular vesicles that has been identified in the circulation of those patients. But next, we were, of course, interested in the capacity of the extracellular vesicle protein landscape and also the lipoprotein particle protein landscape to distinguish those different types of diseases. And therefore, we performed a correlation analysis of the extracellular vesicle profiles, protein profiles that we obtained by mass spectrometry. In this correlation analysis, we could actually observe that within the EV extracts, the correlation is higher within the disease condition, which indicates that there is a specific protein landscape in extracellular vesicle that is associated to a certain disease. And this is what we further confirm by pathway analysis. By contrast, if we look to the LPP extracts, we observe that across clinical conditions that there is no difference or that there is a high correlation between the different disease conditions, meaning that there is no big variation or significant disease-dependent variation in those LPP particles. Furthermore, this correlation analysis confirms that the protein composition of the LPP extracts across the different clinical conditions significantly differs from the protein landscape that we identify in the extracellular vesicle extracts across those clinical conditions. Next, we were also interested to study the dynamics of both the EV and the LPP extracts in serial blood plasma samples. This study was performed on 4 ovarian cancer patients over 5 different time points during their clinical treatment course, starting from diagnosis until the end of the chemotherapy treatment. We again prepared the EV and the LPP extracts by using size and density-based preparation, and we performed a mass spectrometry-based proteomic analysis. This hierarchical clustering again confirms the significant difference in the protein composition between the LPP and the EV extracts, which indicates that the difference between both particle types is more significant than the variation over time within, for example, extracellular vesicles and the lipoprotein particle extracts. But if we again take a look to a correlation matrix of those patients over the different serial blood samples, we observe that the correlation within the LPP extracts is high over the different time points during the patient treatment, indicating that there is no big protein variation over the different time points for this patient. By contrast, if we take a look to the extracellular vesicle extracts, we observe that this correlation between the different time points in the protein composition is much lower. If we indeed take a look, for example, to the set of extracellular vesicle-associated proteins over the clinical time course of those 4 ovarian cancer patients, we observe that time point 1, which is just a blood sample taken prior to surgery, we have a high normalized sum of intensities of those extracellular vesicle-associated proteins, while after surgery, we observe a significant decrease in those extracellular vesicle-associated proteins over the multiple time points. By contrast, in the LPP extract, we don't see this evolution. The reason why there is a decrease in the LPP counts at the first sample is because the sample was just taken prior to surgery and those patients were in a fasting state. So we know that in contrast to the LPP extracts, the EV extracts carry signatures that are disease-specific and that they also show dynamics during the clinical treatment course of the patient. This is what we learn by making use of the fractionation strategy. But by further analyzing all the different fractions of the density gradient centrifugation, there was another set of particles that we identified at a higher density compared to the eukaryotic extracellular vesicles, and that significantly highlighted bacterial hits, both at the protein level but also at the RNA level. Of course, then we started to wondering, what is it? Do we have some bacterial contamination in our sample, or do we maybe identify bacterial extracellular vesicle in the blood plasma of those patients? So what we did is we started off with the assumption, if we do have bacterial extracellular vesicles, they may be derived from gram-negative bacteria and they contain LPS or lipopolysaccharide on their surface. So for each fraction of our fractionation strategy, both for the size exclusion chromatography, but also for the density gradient centrifugation, we compared the different fractions for their LPS content. If we take a look to the different fractions from the size exclusion chromatography column from 1 to 15, we observe a significant peak in LPS in the sec fractions 5 and 6 that correspond to the fractions that elute extracellular vesicles. We also have a second peak of LPS in the later fractions that probably corresponds to HDL associated LPS or soluble LPS. If we then collected the sec fraction 5 and 6 and loaded this on top of the OptiPrep density gradient and screened the different density fractions for the LPS signal, we observed that we have a first peak in LPS in the first fractions of the density gradients that contain the lipoprotein particles. And LPS is indeed known to be neutralized by lipoprotein particles, including HDL and LDL. Secondly, we observed a peak in a density fraction that is much higher than the density where we identify the eukaryotic extracellular vesicles. So this again further supported the assumption that we have bacterial extracellular vesicles. And if we compare this variation in the LPS signal between a patient that has an intestinal barrier dysfunction and a patient without intestinal barrier dysfunction, we observe that although we do not observe a difference in the LPS levels in the lipoprotein particle fraction, we do see a significant difference in the LPS fraction of the density that may correspond to bacterial extracellular vesicles. So to further clarify that we identify bacterial extracellular vesicle at this site in the density gradient, we implemented multiple complementary characterization methods. So first of all, we did a proteomic analysis where we demonstrated the identification of outer membrane proteins, inner membrane proteins, and also cytosolic proteins that are specific to bacterial extracellular vesicles. We quantified the different numbers of bacterial extracellular vesicles making use of the LAL assay that provides endotoxin units based upon LPS, but also making use of the TLR4 to measure the activity levels of LPS, and the TLR2 that identifies LTA. Finally, we performed immune electron microscopy that revealed that we actually identify bacterial extracellular vesicles in the later density fractions of the density gradient. To demonstrate the value of those bacterial extracellular vesicles for biomarker potential, we selected patients with intestinal barrier dysfunction compared to their respective healthy controls. So the patients that we included in this study are HIV patients, inflammatory bowel disease patients, and also patients that were diagnosed with cancer and developed intestinal mucositis during their treatment course. Each time we observed a significant increase in the amount of bacterial extracellular vesicles compared to their respective control conditions. So indicating that in the presence of intestinal barrier dysfunction, vesicles produced by gut microbiota are able to enter the blood plasma of patients. Importantly, and again notifying towards the importance of fractionation of the blood plasma sample, is that other LPS products, meaning that if you did a similar analysis on the crude extract that was obtained by size exclusion chromatography, or a direct measurement on the blood plasma sample, we were not able to distinguish those patients based upon the analysis of the LPS signal. So to conclude, first of all, at the methodological level, I want to refer to the Nature Outlook on extracellular RNA that was published yesterday. And I know my presentation was focused on proteins because there is no time to go into the RNA as well. But we do observe similar results at the RNA level as the protein level. Nevertheless, in red, I indicated some codes that I took from this outlook, and that is that changing a few parameters can really influence the extracellular vesicles that you obtain from a sample. And that's true. That's why it's important to be transparent in the reporting of your material and methods, because it's required to have a reproducibility of the research results that you publish. Secondly, whatever the method, a validated reference material is necessary. And that's also true. Reference material can really help us in understanding pre-analytical factors, but also normalizing data and calibrating instruments. So we have contributed to this by developing the recombinant extracellular vesicles that can assure standardized measurements across multiple applications. And importantly also, and this is true for RNA, but I think it's even more true at the protein level. Although some researchers are successful in profiling extracellular RNA, this bulk approach at the RNA level, but I believe it's also at the protein level, can hide important signals that are of biological relevance. So that's why I think it's really important to implement the fractionation strategy and try to learn as much from one blood sample that we can do, as we demonstrated in this presentation by focusing both on the eukaryotic extracellular vesicles that we identified, but also the lipoprotein particles and the bacterial extracellular vesicles. So going to the conclusions at the biological level, we demonstrate that the biomolecular landscape at the protein level, but also at the RNA level, which is not touched upon in this presentation, is disease specific and is dynamic, but we do not identify a similar approach or a similar variation for the lipoprotein particles. Furthermore, we were able to identify bacterial extracellular vesicles in the blood plasma of patients and demonstrated that their presence in blood plasma associated with intestinal barrier dysfunction. Finally, I want to highlight that since I focused on some methodological aspects, that if you want to learn more on tips and tricks on how to isolate and separate extracellular vesicles, that we have our EMBO practical course where you can learn more about this. Unfortunately, this year's course is postponed until November due to the COVID-19 measures. With that, I would like to thank my collaborators, my team, my lab members, and also the funders to support this research. And then I would like to thank you for attending this EV talk series, and I'm happy to take your questions.
Thank you. Most people are muted, but I'm sure there were a big applause. Thank you very much, An. It's excellent. So what we do now is we ask you all to post questions in the chat area, and I will let you unmute yourself and ask a question verbally. So my question would be too difficult. I have one, so I don't want to do that one first. So I mean, but if you look at plasma, it's the most complicated biofluid we have, right? So how do you compare the differences and similarities of separating vesicles from plasma versus cell culture medium supplemented maybe with exosome depleted plasma or something like that, serum? I mean, so is it much more difficult, you think, than cell culture medium?
I think blood plasma is still much more difficult than cell culture medium. Although we do not implement this approach ourselves, so if we prepare cell culture supernatant, we most of the time use 0.5% EV depleted serum. Just to make sure that the vesicles would still behave like they would do, they can grow, and there is no significant alterations due to serum depletion. but we never really implement like a 10% serum condition if we work on cell culture supernatant. Nevertheless, I would assume that like the lipoprotein particles that you have from LPS, you could be able to separate them by using exactly the same size and density based approach as we do for blood plasma. One of the issues I think then would be that an important step in using cell culture supernatant is that you have a large volume, which you would still have to concentrate before you can load it on a density gradient. And this concentration step can be difficult in the presence of 10% serum. And this is, of course, an issue that you don't have when you study the blood plasma sample.
So I'll ask a question anyway, because, and that is, if you do size exclusion, you know you have your vesicles coming out early, right? And then you get much more protein in relation to the number of particles that you isolate. Now, when we do proteomics analysis of that proportion, the later proportion with a lot of protein and a lot of other contaminating things, we still see a lot of EV proteins there. And like CD63 and membrane proteins. So they're clearly membrane components there. And I'm just a little bit worried that we're losing something.
Yeah, so although I think most of us look at the size and density based method as a bulk analysis of EV, I think we still do, of course, a selection in terms of size and density. Now, why do we selectively? It's a balance you have to find between specificity towards extracellular vesicles and losing some vesicles in return. Because if you would add additional sec fractions and load those on top of a density gradient, the main issue is that you increase the contamination with high density lipoprotein particles. And of course, those float at a similar density as extracellular vesicle, which means that you cannot remove them anymore through using the density gradient. So we, at some point, we have to use a certain cutoff both at size and density, because it's the balance that you make between omitting a few of the vesicles but ensuring that
I mean, the issue I have with this is that the RNA profiles are so fundamentally different, with the slightly higher density components than the lower density components that are floating with the ribosomal RNA peaks, and the high density do not have that. I guess it's what we used to call exosomes and microvesicles 5 years back or 8 years back. And there's no more to be dug into there. I'll let Anna Paulina unmute herself and ask a question, and I'll stop my questioning.
Okay. Hi, Anne. Thank you very much for your presentation. I was wondering if you tried doing the density separation first and after the size exclusion chromatography. And okay.
Yeah.
And the other question is about what was the starting volume that you load into the OptiPrep?
Yeah. So first of all, why do we choose the order of first a size exclusion and then a density gradient? So to be clear, several other groups have reported a size and density based combination, but it's also, I think, interesting to have a discussion on that. So there are multiple approaches. So I think Cecilia Lasser and Jan Lötvall, they published the combination where they do ultracentrifugation followed by a density cushion and size exclusion chromatography. Yet also Edith Buza's group that combines a density gradient with a bind-elute chromatography. So there are definitely multiple approaches to do. So why did we first choose for the size exclusion and then OptiPrep? It's basically because we observed that the viscosity of plasma is quite high and you get a less efficient separation on the density gradient than if you would first implement a sec. So by sec, we first try to remove the HDL particles. Then we load them on a density gradient, and then we remove the lower density lipoprotein particles. HDL particles are also the most abundant particles in the blood plasma, so it seemed to us that it would be most valuable to try to first remove these and then go to the next step. So I presented here also as a size exclusion followed by density gradient, but actually after the density gradient, we do a second size exclusion chromatography, which further removes HDL particles that might still be present in the sample. But the major reason why we also do is to remove the iodixanol from the density gradient fractions, because it can interfere with mass spectrometry-based proteomics, but also with the RNA sequencing. And then with regard to the volume that we load, so for the size exclusion chromatography, we start from 2 ml of plasma. If we collect the sec fractions, it's 2 ml of sec fractions, and we concentrate these to 1 ml, and it's then 1 ml of the crude extract that we load on top of the density gradient.
Thank you.
When you say you concentrate, sorry, when you say you concentrate, you mean you pellet it in the ultracentrifuge, or how did you concentrate?
Concentration is making use of the concentration with the cutoff size of 10 kilodalton. So those membrane concentrators. Yeah.
Okay, thank you very much.
Okay, so we go to Annalisa, please. Unmute yourself and ask a question.
Thank you, Anne, for your great presentation. Very inspiring. I was okay. My first question was the same of Paulina, so that you just answered. So such a complicated way that we need to separate vesicles from other nanoparticles in the blood. How would you translate this in a discovery for? I mean, you're doing this for discovery of novel biomarkers, but how would you do this for in a diagnostic field? How can we see those differences in a laboratory? How could you implement a technique, an easy technique to see those differences, since you show that with other techniques you don't see any difference from patients?
Well, the reason why I'm really a fan of this fractionation strategy, because I'm still convinced that there can be very important biological signals that we will only be able to identify once we have really purified them through their content. So I think you are aiming towards questioning how do you see this in a clinical application. I think it's two different points. This is really discovery, exploring what do we have in the plasma, what are the different particles, how do they differ, how do they evolve during the treatment course, how do they differ between different diseases. So I think it's two different approaches. How I see how we evolve is that from the protein signatures that we establish and the RNA signatures that we make a more dedicated tool, but that's then focused more towards really identifying the extracellular vesicles. So here we explore the biomarker potential and the composition and the dynamics of different types of extracellular particles. If you go to a diagnostic application, you focus on one type or you combine different types, but then you of course need more focused assays, and that can be some surface-based enrichment of certain population of extracellular vesicles. Because I think the next step would still be, we have the lipoprotein particle extracts and we have the EV extracts. The lipoprotein extract still consists of a mixture of HDL, LDL, VLDL, kilomicrons. So you can further scrutinize this preparation and really separate the different lipoprotein particles. And the same you can do for extracellular vesicles, separate small and the large EV from this density fraction and further scrutinize and investigate how the biomarker potential and composition is varying between these subtypes of particles. But that's research and discovery, and it's different from having a clinical application. Yeah.
Okay, thanks.
Thank you. So we'll move over to Hernando. Please unmute yourself and ask a question.
Hello, and thank you for your excellent talk. We work with malaria, and that causes hemolytic anemia. So many times we are very limited with the amount of plasma that we can obtain from patients. I am wondering if you have compared immunoprecipitation of extracellular vesicles with the OptiPrep in proteomics.
Yeah. So combine, whether we compare the density gradient with immunoprecipitation in the proteomics, no, we did not do so because we want to start from the bulk approach. Although we still make a size and density based selection, we did not really compare it with immunoprecipitation. So that's something that we did not do, because then we start immediately to focus on a subtype of extracellular vesicles, while we want to have the broader overview of the different fractions and the biomarker potential of the different fractions. So I do think that immunoprecipitation is a valuable approach, and definitely towards clinical applications will be one of the approaches that will have a high applicability. But I think from the discovery perspective, I like more the bulk format than the immunoprecipitation.
Yeah, I think I agree with you on that, but I'm really looking into the field in terms of functional assays, because this is one of the main problems that we have. For the landscape, I think it's okay, but I think in the long term, we're going to have some kind of very, very specific ways of obtaining EVs from the source that you want to do the functional assays. I don't know if you have really compared functional assays in between your sec and your fractionation.
No. We did. We focused on the biomarkers and not the functional. No, we did not perform functional studies with the different fractions. No.
Okay, and thank you so much.
Thank you. We move on, and it's Rosella Crescitelli.
Yeah. I think that your results about plasma are impressive, honestly, and you really developed a good method. I was wondering if you tried, or if you have any results in comparison with serum, if you did something about serum, vesicles from serum as well. And my second question is, you say that the OptiPrep iodixanol can give side effects when you do mass spec. Because we didn't notice that, or I was just wondering if we just didn't notice, or I don't know, but because we didn't notice this problem with iodixanol and mass spec, which kind of problem did you notice?
Mm-hmm. So first of all the analysis that we did was on blood plasma, so we did not really compare towards serum. Yeah.
You have no opinion about that? I
convinced all my clinicians that we should collect citrate plasma. So they don't want to give me serum anymore.
Okay.
We did not compare with the serum.
Never tried. Okay.
Yeah. But it is an important question, because some people are working with serum and some are working with plasma. I mean, Rosella is in my lab. Actually, we are doing those comparisons and struggling with that. So I don't know if Nasib is on the call, but and there will be information out there soon. But it's really not so easy to. But there's fundamental differences in the profiles of vesicles in plasma and serum.
Yeah, that are not only platelet associated differences.
We should comment on that whenever it's due.
Okay.
Pia Siljander, please unmute yourself and ask your question, and show your camera if you can.
Well, thanks. I already got my answer. So it was this 2 mL of plasma. So that's quite impressive and you can do so much with that. And I also just wrote down that well done with the clinicians, convincing them. Mm-hmm. So and also we should strive to get this message across to the biobanks and the pre-analyticals. So I think there is quite a lot of things we need to do, but a really great talk. Thanks for that, Anne.
Thank you, Pia.
I think we use 6 milliliters usually, correct, Rosella? Yeah, okay, 6 milliliters. We get a lot of information with that. I think it's Ahmed Agad. Please unmute yourself and ask your question. If not, I'll ask it. What do you think about the combination of precipitation method with size exclusion for RNA studies? Can we still trust the specificity of the extracted EVs? Thanks.
So the combination of a precipitation and, is it immunoprecipitation or like?
I think, you know, think PEG, think exoquick, I guess, right?
Exoquick. Well, as far as I'm concerned, if you use a PEG-based approach only, you could as well just look at the total blood plasma. Mm-hmm. If you would combine it with SEC, I think you would still have the similar issues as using size exclusion chromatography only. Yeah? So you will still end up with a mixture of different types of extracellular particles, including EVs, but also like the LPP. So I'm not telling that you cannot do it. That's definitely not the case, that we should all start to do the density gradient, but I think it's just on how you make conclusions from your results that you obtain. If you're in search of really an EV associated biomarker, then you have to demonstrate that it's EV associated. And as you can learn from the MySAF guidelines, it's multiple ways that you can achieve this. You can further purify the vesicles and do a density gradient, but you can also combine it with protease, RNase treatments, with or without the detergent treatment, to really identify whether the RNA that you have is associated with a vesicle structure or not. So there are definitely multiple approaches. The key point that I wanted to make with the presentation is that we can discover a lot by fractionating and separating different types of particles. But if so
that's a different question, right? If you have a pure RNA question, you could just do RNA extraction, right? If you want to know what is in the vesicles or elsewhere, you need to do the separations.
Yeah.
I just want to comment to say that in our hands, most of the plasma RNA is in the vesicle fraction. But I think other people need to confirm that or dispute that before we can state that with 100% certainty. But really, we see most of the RNA in the vesicle fraction. So, Navnet Dogra is on board here, and please unmute yourself. And the interesting thing with Navnet, he's got technologies which potentially could isolate those small vesicles from proteins.
Yeah. Hi, and that was my question, actually a comment. That so we isolated serum vesicles, and we isolated vesicles of about 70 nanometers and about 170 nanometers precisely. And the 70 nanometer vesicles do not have platelet RNA, but the 150, 170 nanometer vesicles did have platelet RNA. So that was what you guys were discussing. But my question that I wrote was, anyways, we have to do centrifugation, small 300G and then 2000G. And we are creating some fragmented membrane vesicles. Are those the vesicles that are coming, small particles, 20, 30 nanometer particles that are coming later in the SCC fraction, which still have CDs, but they don't seem like vesicles?
I think it's a general observation that especially like the tetraspanin protein are really across multiple fractions in the size exclusion chromatography column. Well, I think it's time to do an in-depth electron microscopy analysis of all the sec fractions, I would think, to really identify whether we still have fractions. An alternative strategy would still be to do the RNase, protease without detergent treatment on the different sec fractions and see where we end up with, if we still end up with vesicles, and if it are still vesicular structures, are it indeed fragments that regenerated, or do we have really small EVs in plasma that we are indeed excluding? It's an important study to move forward with, I think.
I think I'm quite impressed because Nanette is in New York City, so that's the middle of the night. Right?
I am, and I'm up at very early morning.
Yeah, thank you, thank you, Anne, for attracting that attention. Just checking here. So, Antonella Bongiovanni, please. If not, I will ask the question. Do you think envelope viral particles, like coronavirus, are isolated together with EVs following SEC and density gradient?
Who knows?
Yeah, I have made a whole blog with Ken Whitwer, two of them actually, about similarities and differences and distinctions between SARS-CoV-2 virus and extracellular vesicles. And there's certainly a lot to learn about that. It is an envelope virus, but it has membrane components. But where does it end up in the density gradient? I don't actually know.
Well, the only thing I know is that if we have like the virus-like particles, the recombinant extracellular vesicles that we prepare that have the HIV-1 GAG polyprotein, is that if we implement the density gradient that I presented here, so the top-down density gradient, flotation-based, that we have those vesicles in the same density fraction. However, if you implement the velocity-based density gradient, they do fraction to different density fractions. They do flow to different density fractions. So that's because they have very tiny differences in their density that you cannot identify by making use of the flotation density gradient, but you see those differences with the velocity density gradient. And it's also the approach that has been proposed to distinguish HIV viral particles from extracellular vesicles. So I would assume to implement this strategy.
So top loaded, basically, that's what you mean? Or bottom loaded?
Well, the velocity gradient, what we did is, top loaded as well, yeah.
Nader?
Hi, it was a really nice talk, Anna. I have actually two questions. The first question is about, do you try to detect the gram-positive derived vesicles? And the second question, if I understand correctly, I mean, even in healthy people, you still can detect some bacterial EVs. Are they I think they are really low in concentration, but how did they end up in plasma?
So the question is related to the bacterial, if I did not completely understand, but to the bacterial extracellular vesicles, how in a healthy...
gram positive. So Staphylococcus vesicles, basically, right?
Yeah, so we indeed identify both gram-positive and gram-negative bacterial extracellular vesicles. Compared to the eukaryotic extracellular vesicles, their concentration is like 100 to 1000-fold lower, so it's even more challenging to study those vesicles. But how do they end up with the healthy donors? First of all, the concentrations of bacterial extracellular vesicles that we identify in healthy donors are really, really, really low. They are close to the detection limit of the quantification assays that we use. And it's really much higher in patients that have leaky gut symptoms. So it's... I'm not really convinced that we have an abundant presence of bacterial extracellular vesicles in healthy donors unless maybe they had a very large alcohol consumption just before the blood draw, because it also impacts the leaky gut. But I don't think in general that there are high levels of bacterial extracellular vesicles in healthy donors. And it was also what you could see in the quantification compared to the HIV, inflammatory bowel disease, and the cancer patients as well.
Yeah, thanks.
So the next question is really important, actually, from Rosella came back, and that's about density gradient and the type of density gradient that we'll be using. We talked about this before during this talk, which is sucrose gradient is good at separating vesicles and in different densities, but a lot of experience tells us that they cannot be used functionally after that. You can't put them in a cell system and expect a decent function, whereas when you do that with iodixanol density gradient isolated vesicles, it works. Iodixanol, everybody should know, it's a clinically approved contrast medium, so it's not strange to use that. And Rosella's question is really important there. What issues do you see with the iodixanol in mass spec and RNA analysis?
I wrote it down that I still had to answer that question. So the issues that we have, or what we at least noted, but I think you maybe don't notice it because your approach is different. You first do a gradient and then a size exclusion chromatography. So you remove the iodixanol anyway from the...
No, from the proteomics that I did, but was about tissue, I just did the gradient, iodixanol gradient, and sent samples to proteomics, adding, they just asked me to add SDS.
Mm-hmm. Okay.
And they did the analysis, and I guess that we always perform proteomics in this way, telling us that we should not have any issue, and we didn't notice any issue. This is why I'm asking, but it's important if it can give problem, of course.
But it's indeed not an issue if you have an abundant sample. So, for example, if you prepare cell culture EVs, and I assume it's similar for tissue EVs, you have a much higher amount of input sample for the proteomics analysis. Then we also do not observe an interference with iodixanol. However, if you have a low amount of sample, which is what we most of the time have if we isolate vesicles from blood plasma samples, then we do observe that there is an interference with iodixanol. And how is this interference translated? You have almost no peptide hits. So it's really, it really hampers the identification of proteins, but also RNA. So we had the same issue with RNA, and we were also surprised in the beginning because we optimized everything with cell culture EV and spiked samples, but then of course you have high amounts of input material, and that is not the case, especially, for example, if you separate bacterial extracellular vesicles or extracellular vesicles from healthy donor samples, you have a limited input amount, and it's most for those samples that it's an issue.
But so it means that maybe the problem, it can be connected only because the sample is too diluted, not because iodixanol gives problems by itself.
No,
it can be
really,
this.
No, it's really the iodixanol.
Okay.
Yeah, because
so if you do it with sucrose, you don't have the issue?
Well, we didn't try with sucrose, but if we added increasing concentrations of iodixanol to the same starting sample, it was from a certain concentration of iodixanol contamination that you have a significant reduction in the number of proteins and RNAs that were identified. So, and you also observed this characteristic iodixanol profile on the mass spec spectra. So it's really a iodixanol issue. Yeah, but for low abundant samples. So if you have enough starting material, it's indeed
because I never had this problem, but good to know.
Yeah.
So I'm enjoying this dialogue so much that I'm going to continue with a very big question, if you don't mind. And it's an issue that arises from all the work we've done for the last 15 years, but also as my role as editor-in-chief for GEV. And we get a lot of papers submitted that are doing different ways of eliminating fetal calf serum or bovine serum vesicles from their cell cultures. And either they use EV depleted serum, or they do serum depletion, and they do that in different ways. So, Caroline, I think we could have a whole session about this only, right? With 3 or 4 different speakers, 2.5 hours, and then try to figure this out. And we're struggling to understand what is the appropriate way of doing this. I'm thinking about, of course, cell culture experiments now, not necessarily biofluid or not biofluidization. What's your thinking around that big issue?
So how to deplete your EVs from serum that you use for cell culture?
Yeah, so basically you either do that or you deplete the serum and grow yourself, right? Grow yourself. So I'm not sure how much you've tried that, but in our hands that changes the EV profile totally. So
well, we did not really make those comparisons. So what we do use, so for example, to produce recombinant extracellular vesicles, we do avoid the use of serum, and therefore we make use of Optimem, but hectonitrate T cells are quite, let's say, they can handle some conditions, but in Optimem they still maintain their normal morphology and their growth characteristics. So I think that's definitely one of the conditions that we can consider, that we can make use of media that do not contain serum. But of course, then you have those publications that do show that there is some impact of those media on the EVs that you identify. So I think it's a really big issue. And as far as I'm concerned, as long as you transparently report what you did, I think, yeah, I cannot solve this question in this discussion. I think we need some experimentation.
No, I don't expect you to. I just wanted to throw out that big question out there because to make people think about it as well, because it is something I think we need to come back to. Thanks. Thank you, Ann. This was great. Beauty with these things is we can continue until we're fed up or we're exhausted. And it's 1 hour and 20 minutes now, so that was really enjoyable. Carolina, thank you.
Yeah, thank you very much, and that was really wonderful. And you definitely developed a very nice separation method and characterized them, and showing the difference between LDL and EVs very clearly. I think that was very nice work, and also looking at the difference in the bacterial EV as well. And I'm really impressed with the reference material. And I was actually wondering whether that is available. Can we actually have it? When are you going to actually distribute that reference material and share it? I think that will be it will be very useful, I guess, for the community. So,
So it was also commented on in the Nature Outlook on the xRNA. So I get a lot of questions for the reference material, so we can definitely already distribute it, but it has been exclusively licensed, and the company is on its way to produce it and to have all the quality controls that we give them in place. So normally, I don't know, of course, given the COVID-19 measures, how it has delayed their production process, but normally it should be available in the second half of this year.
Okay, that's great. We're looking forward for that.
If you have an urgent need, we can always set up an MTA agreement to ship some materials.
Awesome. I will chase that up. So and thanks very much for the interesting discussion that you lead. That was awesome. I think I like to comment about that serum free or not, because in our hands, like this, the presence of serum is very essential because the cells died without it. So we have to have serum no matter what. So I guess it's probably case to case, and agree with Anne, as long we report it clearly what we have done. I think hopefully that is acceptable. Yeah. Well, I think with that, we conclude the meeting, and